Assalamu alaikum. My name is Abi Waqas, and now we will discuss about the cell membrane of bacteria. Let's talk about the structure of the cell membrane. The structure of the bacterial plasma membrane is that of unit membrane, that a fluid phospholipid bilayer composed of phospholipid 40% and peripheral and integral protein 60% molecule. The phospholipid of bacterial cell membrane do not contain sterol as in eukaryotic, but instead consist of saturated or monosaturated fatty acid, rarely polyunsaturated fatty acid. Many bacteria contain sterol-like molecule called hoponoids. The hoponoid most likely stabilize the bacterial cytoplasm membrane. The phospholipids are amphoteric molecule with a polar hydrophilic glycerol, a head, a tail, via an ester bond to two nonpolar hydrophobic fatty acid tails. The phospholipid bilayer is arranged such that the polar ends of molecule form the outermost and innermost surface of the membrane, while the nonpolar end form the center of the membrane. So here is the structure of phospholipid bilayer. Let's talk about the fluid mosaic model. The plasma membrane contains protein, sugar, and other lipid in addition to the phospholipid. The model that describes the arrangement of these substances in lipid bilayer is called the fluid. Dispersed within the bilayer are various structural and enzymatic proteins which carry out most membranous function. Submembrane proteins are located and function on one side or another of the membrane, peripheral protein. Other proteins are partly inserted into the membrane or possibly even traverse the membrane as channel from the outside to the inside. Integral proteins. Lipids are capable of rapid diffusion within the layer but flip-flopping from one layer to another. So here is the structure of fluid mosaic model. Let's talk about the function of the cell membrane. The cytoplasmic membrane is selectively permeable membrane and acts as an osmotic or permeability barrier. Location of transport system from specific solute, nutrients and ions. Energy generating function, respiratory and photosynthetic electron transport system, proton motive force and transmembrane ITP synthesizing ATPase. Synthesis of phospholipid including membrane lipid and lipopolysaccharide of gram-negative cells. Synthesis of murin both in growing cell wall and in the transverse septum that divide the bacterium during bacterial division. Assembly and secretion of extra cytoplasmic protein. So the coordination of DNA replication and segregation with septum formation during binary fission, site of the insertion for flagella, chemotaxis, motility, Percy and sensing function. Waste removal, formation of endospore beside transverse protein that selectively mediate the passage of the substance into and out of the cell. Prokaryotic membrane may contain sensing protein that mere concentration of molecule in the environment or binding protein that translocate.